hello friends, hello G, it's your boy again, falling with a star. So guys, today our topic is fructose metabolism. Let's look at some facts about fructose. Fructose is a 6-carbon ketose sugar. You see, as a monosaccharide, you can either have the functional group aldehyde and you become an aldose, or the functional group ketone and you become what? A ketose. So fructose is what? A ketose sugar. Some people also say that fructose is a what? A monosaccharide. It can be obtained from honey, corn syrup, and also fruits. It cannot be produced by the body, unlike galactose. Galactose can be produced by the body by the breakdown of what? Glycoproteins and also glycolipids. It is an isomer of glucose. Glucose is an aldose sugar, whilst fructose is what? A ketose sugar. It is a component of sucrose. Sucrose is a disaccharide, and whenever sucrose is being broken down by sucrase, we get glucose and fructose. Now guys, let's look at the difference between the absorption of what? Fructose versus the absorption of what? Glucose. Fructose, let's say that you've taken in this fruit, it contains fructose. Fructose will be absorbed into the intestinal mucosa cells, but this is the case that fructose is a large compound and cannot enter into the cell by simple diffusion. So it needs a channel, and this channel is called glucose transporter 5. We actually have 14 types of these glucose transporters. And let's look at some features of this glucose transporter 5. It is bidirectional, meaning fructose can use the same channel to enter into the cell and also use the same channel to come out of the cell. It is insulin independent, unlike GLUT4, which is insulin dependent. It needs insulin to bind to a receptor on the cell to cause it to open. But this GLUT5 is insulin but independent. It is also the only transporter that transports only fructose, unlike the other glucose transporters which can transport glucose, fructose, and galactose. This transporter only transports what? Fructose. It doesn't depend on sodium gradient. You understand this when I talk about glucose absorption. The transport of fructose into the intestinal mucosa cell is by what? Facilitated diffusion. So guys, let's look at that of what? Glucose. Glucose is also a large compound and needs a channel. So it needs this channel, sodium-dependent glucose transporter 1, to get into the intestinal mucosa cells. We have two types of this sodium-dependent glucose transporter. We have sodium glucose transporter 1, which can be found in the enterocytes, which is the intestinal mucosa cells, and sodium glucose transporter 2, which can be found in the proximal convoluted tubule. These sodium glucose transporters, they are unidirectional. That means glucose can only use them to get into the cell. It also depends on sodium. Sodium needs to move down its concentration gradient to help the glucose to also move into the cell. So this transport is actually by secondary active transports. So guys, now this is the case that fructolysis mostly, or fructose metabolism mainly occur in the liver. So we need to transport the fructose from the intestinal mucosa cells into the liver. And you know that the function of the blood is to transport what? Substances in the body. So this fructose will get into the blood. It will leave the intestinal mucosa cells by this channel we call GLUT2 and get directly into the blood. And this blood will transport the fructose into the what? Into the liver. So it will enter into the liver cells, and the cells too will not allow the fructose to get into it by simple diffusion. So fructose needs a channel to get into the what? The cells of the liver, and that channel is called what? GLUT2, and it is also insulin independent and also bidirectional. So in the liver cells, that's where fructolysis occur, or fructose metabolism is going to occur. So guys, let's look at fructose metabolism in the liver. So guys, how fructose is being metabolized in the liver is actually different from how fructose is being metabolized in the adipocytes or in the skeletal muscles. In the muscles or the adipocytes, this fructose, by the help of the enzyme hexokinase, is being converted to what? Fructose 6-phosphate. And depending on the body state, this fructose 6-phosphate can be used in glycolysis or in the synthesis of what? Glycogen through the process of glycogenesis, where fructose 6-phosphate will be converted to what? Glucose 6-phosphate, and glucose 6-phosphate into what? Glucose 1-phosphate, which will be used in the synthesis of what? Glycogen. Or this fructose 6-phosphate can be used to actually generate what? Glucose through the process we call what? Gluconeogenesis, where the fructose 6-phosphate will undergo mutation into glucose 6-phosphate, and by the help of the enzyme glucose 6-phosphatase, we get what? Glucose. So now let's look at how the fructose is being metabolized in the liver. In the liver, fructose, by the help of the enzyme fructokinase, is being converted to what? Fructose 1-phosphate. And this process actually requires ATP, energy in the form of what? ATP. And let's look at some features about fructokinase. This fructokinase is solely for fructose. It has high affinity for fructose, and biochemists will say, in other words, it has what? Low Km. It is this step, the conversion of fructose to what? Fructose 1-phosphate, is the rate limiting step or the committed step in what? In fructolysis. And this fructokinase is mainly being found either in the liver, the kidney, or the intestinal mucosa cells. After we've gotten this fructose 1-phosphate, this fructose 1-phosphate, by the help of aldolase B, can be cleaved into these 2,3 carbon molecules, dihydroacetone phosphates and glyceraldehyde. Let's focus on the dihydroacetone phosphate. Depending on the body state, this dihydroacetone phosphate is a glycolytic intermediate and can be used in what? Glycolysis, to gain what? Energy. Or this dihydroacetone phosphate can actually undergo the reverse of what? Glycolysis, which is what? Gluconeogenesis, for us to what? Obtain what? Or generate what? Glucose. This dihydroacetone phosphate can also be converted to what? Glycerol 3-phosphate by the help of the enzyme glycerol 3-phosphate dehydrogenase. And this glycerol 3-phosphate can be used in the synthesis of what? Triacyl glycerol. Can be used in the synthesis of what? Triacyl glycerol. Now let's focus on the glyceraldehyde. This glyceraldehyde, by the help of the enzyme, or let me say that this glyceraldehyde, depending on the body state, it can be converted to what? Glyceraldehyde 3-phosphate by the enzyme triose kinase. And this glyceraldehyde 3-phosphate, depending on the body state, can be used. You know that this is a glycolytic intermediate, so it can be used in glycolysis, or depending on the body state, it can be used in what? Gluconeogenesis, during the starvation or the fasting state. This glyceraldehyde can also be converted to glycerol by the help of the enzyme alcohol dehydrogenase. And this glycerol, by the help of the enzyme glycerol kinase, can be converted to what? Glycerol 3-phosphate. And I said that this glycerol 3-phosphate can be used in the synthesis of what? Triacyl glycerol. I said that this fructose 1-phosphate can be cleaved by what? Aldolase B. We have 3 types of aldolases in our body. We have aldolase A, aldolase B, and aldolase C. Aldolase C is being found in the brain. Aldolase B is being found mainly in the liver, kidney, and also the intestinal mucosa cells. Aldolase A, which is common in all tissues, but mainly can be found in the what? In the skeletal muscles. Now let's look at how this triacyl glycerol is being synthesized. You see, in glycolysis, the end product is what? Pyruvate. And pyruvate can be converted to what? Acetyl-CoA. And this acetyl-CoA can be used to synthesize what? Fatty acid. And glycerol produced here plus 3 fatty acids help us to get what? The triacyl glycerol. So we say that high fructose metabolism is related to what? Obesity, because high fructose metabolism can lead to what? High levels of what? Triacyl glycerol in our body. So guys, let's look at fructolysis versus what? Glycolysis. You see, fructose is being converted to what? Fructose 1-phosphate by the help of the enzyme aldolase B, cleaved into what? Dihydroacetyl phosphate, or dihydroacetyl phosphate, and what? Glyceraldehyde. Dihydroacetone phosphate and what? Glyceraldehyde. And this glucose is being converted to what? Glucose 6-phosphate in the liver by glucokinase, or in other tissues by hexokinase. And this glucose 6-phosphate is being converted to what? Fructose 6-phosphate, which will be converted to what? Fructose 1,6-bisphosphate by the help of the enzyme phosphofructokinase 1. And this step in glycolysis is known to be the what? The rate limiting step. And this fructose 1,6-bisphosphate will be cleaved by either aldolase A, B, or C into what? Dihydroacetone phosphate and glyceraldehyde 3-phosphate. You can see that in fructolysis, this rate limiting step in glycolysis is being what? Is being bypassed. So we say that the fructose metabolism is very rapid than glucose metabolism because it bypassed the rate limiting step in glycolysis. So guys, let's look at fructose metabolism disorders and also the sorbitol pathway. So guys, the last 2 topics are fructose metabolism disorders and what? The sorbitol or the polio what? Pathway. So let's look at the fructose metabolism disorders. So this is the pathway fructose is being converted to what fructose 1-phosphate, which will be cleaved to what dihydroacetone phosphate and what glyceraldehyde. So these are the two key enzymes in what fructose metabolism. So in the absence of fructokinase, what happens? You see, fructose will enter into the liver cells by using this channel we call GLUT2, which is bidirectional. And the addition of phosphate to fructose is to trap fructose in the liver cells. You understand? So if in the absence of fructokinase, we are not able to add the phosphate to the fructose, it means that we will not be able to trap the fructose in the liver cells. So the fructose can also leave the liver cells by using this same channel, because it is what? Bidirectional. And when this fructose leave this liver cells, it will go directly into the blood. So there will be high accumulation of fructose in the blood, and this will actually exceed the renal threshold and will appear in our urine. This is why this disorder is called essential fructosuria. And this is also a hereditary disorder. Inheritance is autosomal recessive, and it's also a benign asymptomatic condition. No treatment is actually indicated. What you can do is to stay away from fructose, or eat fructose-free diets. So now, in the absence of aldolase B, what actually happens? You see, this fructose will be converted to fructose 1-phosphate, and ATP is required in this step. And this is the case that we cannot metabolize fructose above this step because of the absence of aldolase B, meaning that we are going to waste ATP. You understand? And ATP here will become depleted, and ATP will be converted to ADP, and this we will use this ADP. So this ADP will also be converted to AMP, and when this AMP is being broken down by this enzyme we call AMP diaminase, we get uric acid. And this uric acid is very toxic to our body. It can lead to hyperuricemia or also lactic acidosis. So high levels of uric acid can lead to hyperuricemia and also lactic acidosis. And this is the case that the depletion of ATP means that depletion of inorganic phosphates, and the wastage of inorganic phosphates, which leads to its depletion, is what we call the sequestering of phosphates. And one thing is that this inorganic phosphate is also being required by glycogen phosphorylase to break down glycogen for us to get glucose. And it's also being required by gluconeogenic pathway for us to get glucose. So in the absence or in the low level of this inorganic phosphate can actually lead to hypoglycemia, because we cannot get glucose from glycogenolysis, cannot get glucose from gluconeogenesis. So it will lead to hypoglycemia, and this can also lead to vomiting, hyperuricemia, which I have already talked about, jaundice, and also hepatomegaly. So guys, let's look at the sorbitol or the polio pathway. This sorbitol is a polyhydric sugar alcohol. We can also call it polio. That's why this pathway is also called the polio pathway. And this sorbitol pathway is a way glucose converts itself to fructose when glucose is in excess. As for example, in diabetes mellitus state, we have excess glucose in the blood, and some of these glucose will try to convert themselves to fructose, and sorbitol is the intermediate in this pathway. So in excess glucose, this will actually activate a certain enzyme we call aldose reductase. When glucose is in excess, that is when this aldose reductase becomes activated. And this aldose reductase, because it becomes only activated in excess glucose, we say that it has high Km or low affinity for glucose. This aldose reductase will actually reduce the glucose to sorbitol. And in this step, NADPH is being required. So NADPH will be broken down to NADP plus and hydrogen ion. This sorbitol, by the help of the enzyme sorbitol dehydrogenase, is being converted to or is being oxidized into fructose, and in this step NADH is being required. But this is the case that this aldose reductase, alright, it can be found in organs like the eyes, the lungs, the kidneys, the nervous tissues, the seminal vesicles, but it is absent in the liver. Because aldose reductase is absent in the liver, we say that this sorbitol pathway is absent in the liver because liver doesn't have aldose reductase. But the sorbitol dehydrogenase can only be found in the liver and seminal vesicles. These organs lack this sorbitol dehydrogenase. So after the conversion of glucose to what? The sorbitol cannot be oxidized to fructose. So this sorbitol will be accumulated in these organs. So this sorbitol will become what? Accumulated in these organs. And one thing about sorbitol is that it has a high osmolarity, meaning that it draws water closer to itself. And because of that, this sorbitol accumulation can lead to what? Cataracts in the eyes, in the nervous tissues. It can lead to what? Neuropathy. In the kidney, it can lead to what? Nephropathy. So some people say that the pathological changes in diabetes mellitus is as a result of this what? Sorbitol pathway. So guys, that's the end. And if you haven't subscribed to this channel, please subscribe to this channel. Nice. Thank you. Bye.